Now we move on to this interesting cycle, that is the Kelvin cycle, or we also call it as C3 cycle. Now where does this C3 come from? C, carbon, 3 carbon. So something like that. You'll understand the meaning C3 cycle once you are done with this Kelvin cycle, once you understand this Kelvin cycle. So Kelvin and Benson, they discovered the path of carbon in this process. So first stable compound of the Kelvin cycle is 3-CpGa, that is the phosphoglyceric acid, which is a 3 carbon compound, and thus the Kelvin cycle is also called as the C3 cycle. Got the answer why it's called as C3 cycle? So it was studied by Kelvin on chlorella and scanidasmus. These are the microscopic unicellular algae, and they can be easily maintained in the laboratory, right? Now during this experiment, Kelvin used chromatography technique and the radioactive tracer. He used a carbon isotope, that is C14 technique, for detecting the reactions of the C3 cycle. So he had 3 isotopes of carbon, C12, C13, C14. Okay, so here 6 protons, 6 neutrons, 6 protons, 7 neutrons, 6 proton, 8 neutrons. So that makes 12, 13, 14 respectively. Now Rubisco, one more thing that you need to understand here is Rubisco. Ribulose bisphosphate carboxylase oxygenase is the main enzyme in this C3 cycle. Now it's always present in the stroma, and it makes about 16% of the protein of the chloroplast. So it's an important enzyme. So Rubisco is the most abundant enzyme on earth also. Think how. So carbon dioxide acceptor in the C3 cycle is the ribulose 1,5-diphosphate, that is RUBP, and this carboxylation reaction is catalyzed by Rubisco. Understood the relation? The ribulose 1,5-diphosphate RUBP, which is there, which is the carbon dioxide acceptor. Here in the carboxylation reaction, it is catalyzed by Rubisco. Okay, so this is the Rubisco, RUBP, photorespiration. So in the Kelvin cycle, the low temperature, high carbon dioxide to oxygen ratio, Kelvin cycle, and here oxygen, high temperature, low carbon dioxide is to oxygen ratio, photorespiration. Also Atriplex hastata and A petula are the temperature species which are the C3 plants. So we got some help from there. Now 6 turns of the carbon cycle, that's the Kelvin cycle, 6 turns of the Kelvin cycle are required for the formation of one glucose molecule. You know that a glucose molecule has 6 carbon, right? So 6 turns of carbon cycle are required for the formation of one glucose, meaning every cycle is yielding you 1 carbon. So let us go over the stages very carefully here. So stage 1, carbon fixation. Stage 2, reduction. Stage 3, regeneration of the starting molecule. Right? So in these 3 cycles, these 3 stages, the entire thing is happening. So you see over here, 3 molecules of carbon dioxide Rubisco undergo 6 molecules which falls into 2 sets, a pair, that's 3 PGA. Here the ATP converts to ADP. Okay, then another conversion, 6 NADPH converting to 6 NADP plus 6P. Here you get 6 molecules of G3P. Okay, so then you see 1 molecule of G3P, like half molecule of glucose, and 5 molecules of G3P that's moved on. So you see the regeneration of the starting molecule over here. Again, liberation of energy. Rubisco comes into picture. So this is the whole cycle. So in this cycle, for the formation of one molecule of the hexose sugar, glucose, 18 ATP and 12 NADPH are used. Remember this calculation. Remember this thing. That is 18 ATP and 12 NADPH2 are used. Students, this is very important question. So here we land up onto this question, and the question is, fixation of one carbon dioxide molecule through Kelvin cycle requires what? 1 ATP and 2 NADPH2, 2 ATP and 2 NADPH2, 3 ATP and 2 NADPH2, or is it 2 ATP and 1 NADPH2? What do you think is the answer? So here the answer is C, 3 ATP and 2 NADPH2. Fixation of one carbon dioxide molecule through Kelvin cycle requires 3 ATP and 2 NADPH2. Now let's see this whole of the Kelvin cycle, the C3 cycle, and understand it well. So Kelvin cycle is divided into three distinct phases: carboxylation, glycolytic, reversal, and regeneration of the RUBP. So three different phases that you must remember. Carboxylation is one, glycolytic reversal is another, and regeneration of RUBP is a third one. Now here, talking about the first one, before that we land upon to another question, and the question is, in C3 plant, the first stable product of photosynthesis during the dark reaction is malic acid, oxaloacetate acid, 3-phosphoglyceric acid, or is it phosphoglyceraldehyde? And you know that in C3 plants, the first stable product of photosynthesis during the dark reaction is C, 3-phosphoglyceric acid. Now we move on to understanding the three phases that are there. There is the first phase, that is carboxylation. Here you see 6 molecules of RUBP when come with the 6 molecules of carbon dioxide give you under the presence of Rubisco and carboxyl dismutase. You see 6 carbon unstable compound. You give 12 molecules of 3-phosphoglyceric acid, right? So here this is the second step, that is a glycolytic reversal. Here you see 12 molecules of 3 PGA plus 12 ATP in the presence of a triokinase gives you 12 molecules of 3-bisphosphoglyceric acid, 1,3-BIPGA. Okay, further dehydrogenase takes place, and you see 12 molecules of the 3-phosphoglyceraldehyde, 3-PGAL triose phosphate plus 12 H3PO4 is formed. Now here in glycolytic reversal, out of the 12 molecules, 2 molecules go to synthesize the sugar, starch and the other carbohydrates, and the remaining 10 molecules are recycled to regenerate the 6 molecules of ribulose 5-phosphate after a series of complex reactions. Now out of the 2 molecules of the PGAL, 1 molecule is converted to its isomer 3-hydroxyacetone phosphate. Okay. So here in the glycolytic reversal, 1 molecule of the PGAL plus 1 molecule of the DHAP in the presence of adolase gives you 1 molecule of fructose 1,6-bisphosphate biphosphate, which is 18 carbon, and 1 molecule of fructose, which is isomerase enzyme. In the presence of isomerase enzyme gives you glucose, which further can convert to sucrose or starch. Now moving on to the third phase, that is the regeneration of the ribulose 1,5-bisphosphate or biphosphate. Here 2 molecules of the fructose and 2 molecules of PGAL. Here this is 12 carbon, this is 6 carbon, and under the presence of a trans ketolase gives you 2 molecules of erythrose, that's 8 carbon, plus 2 molecules of xylulose, that is 10 carbon. So yeah, 12 plus 6 equals 8 plus 10. Yeah. So further you see that 2 molecules of erythrose plus 2 molecules of DHAP under the presence of aldolase gives you 2 molecules of pseudoheptulose 1,5-BIP. And further you see 2 molecules of pseudoheptulose plus 2 molecules of PgAl, and the presence of trans-ketolase gives you 2 molecules of xylulose and 2 molecules of ribose. And further you see that 2 times 2 molecules of xylulose in the presence of epimerase gives you 4 molecules of ribulase, that's 20 carbon. And further you see that 2 molecules of ribulase and in the presence of isomerase give you 2 molecules of ribulose. And further you see 6 molecules of ribulose plus 6 ATP, and in the presence of kinase gives you 6 molecules of ribulose, 1,5 BIP, CO2 acceptor, and 6 ADP. So that was all about the Kelvin cycle.